before. Hello, Jan. Can you hear me? Dr. Jan, are you here? I think the speaker is there. Dr. Jan, can you hear me?
Yes.
Oh, that's lovely. Cool. So, do you think I can still say good morning to you guys?
杨院长可以开始吗？您那个摄像，您那个话筒可以打开。
哎，可以哈，可以吗
Yes, then here we can start. 他会介绍一下您，然后您再开始哈。
好的。
I see.
完整画面看到吗？
Will I want to start the second session? I welcome all you guys.
先生吧，先生。
Second day of the symposium, second session. In this session, we have three eminent speakers.
你聊生活那个密切向上的，
Two speakers.
他们那个
So we have total three speakers in this session. Two eminent speakers from the online, from the channel, and one from the offline from over here. So I welcome all three speakers and the audience in this session. The first speaker, if you can hear me, let me introduce. Can you hear me? So let me introduce Dr. Pinjit Jan, the professor of chemistry, and he has over 85 international publications in reputed journals. The most promising factor of his achievement of he has more than four scientific and technological progress awards. So this is the award which simply reflects his achievement in the scientific area which he has done in the last couple of years. Currently, he is a professor, and he is going to talk about the progress of that acute capsule in prevention and treatment of stroke. So, this is over to you.
杨院长，麻烦您打开话筒，然后您可以开始了。
好了，各位专家、各位同仁，大家下午好。我是西南医科大学中西结合学院附属中医院的杨思进哈。下面呢，我给大家分享智能合约通力胶囊防治脑血管疾病的研究进展。我将分为以下4个方面给大家分享。第一个方面，智能合约通力胶囊的一些概述。智能合约通力胶囊是杨思进教授在20多年临床诊疗的基础上，以重用补气、辅以化产通络、佐以祛风为立法指导研发的纯中药的这个院内制剂。该药于2003年获得了泸州市药监局批准的医院制剂批号。这个产品呢，具有组方合理、药材到底、工艺优良的特点。它的理论基础啊，包括治血先治风，风去血自通，风药开旋理论。智能合约通胶囊组方和功能主治，包括它的治法呢，就是一气祛风，通络开窍，通络开旋。主治用于缺血性脑血管疾病、冠心病、静脉血栓性疾病。成分主要有黄芪、水蛭、地龙、大血藤和桂枝。那么我们这个几个药品呢，君药是黄芪，成药是水蛭，佐药包括大血藤和地龙，使药是桂枝。我们这几个药品呢联合作用下形成了祛风开旋的作用。那么智能合约通胶囊呢，基础研究方面呢，我们做了自己工艺的研究、毒理的研究和药效学的研究。自己工艺研究包括生产工艺的研究。我们 做了2种工艺的对照一个是旧工艺和第二个是新工艺这2种工艺的对照。那么2种工艺对照的结果呢，显示新旧工艺前期药效学实验比较2者没有明显的差别，但是新工艺能够节约中药饮片和药用的辅料，新工艺还有更稳定、标准、更完善。我们自己工艺研究还包括质量标准的研究还有这个质量标准研究的结果显示提供了定性和定量的检测方法。从独立研究方面，我们进行了集毒试验和长毒试验。集毒试验显示智能活跃通胶囊对受试动物未见明显的急性毒性作用，表明该药具有良好的安全性。长期毒性实验表明我们这个智能活跃通胶囊对受试动物未见明显的长期毒性作用，表明该药具有良好的安全性。从药效学研究方面，我们做了以下系列的研究。研究1：智能活跃通胶囊对脑出血大鼠血清烟性因子的影响。那么研究结果发现，智能活跃通胶囊能有效减轻脑出血后脑水肿，该作用可能与降低血清烟性因子水平有关。研究方向2：智能活跃通胶囊对模型大鼠脑出血后脑组织水肿及闭锁连接蛋白1和封闭蛋白的影响。结论后发现，智能活跃通胶囊能明显改善大鼠的脑出血后脑水肿。那么这个机制呢，可能与上调脑组织的闭锁连接蛋白1和封闭蛋白 mRNA 的表达有关。研究方向3：智能活跃通胶囊通过 ROCK 通路改善脑出血后脑水肿的实验研究。研究结果发现，智能活跃通胶囊可能通过抑制 ROCK 通路调节血脑屏障功能，改善脑出血后脑水肿。研究方向4：智能活跃通胶囊对
चल रहा है चल रहा है हाँ आप यहीं से बोल देंगे क्या आवाज नहीं आ रही है इनकी
छोड़े जा रहे हैं कोई में
翟院长这边听不到声音了，然后幻灯片也没有，也停止分享了，是断网了吗？严院长您在吗？严院长在吗？严院长在吗？ maybe we can start next. Looks like there's some technical issue. Dr. Zahi?
Since we have some plenty of time, it's possible for you just to briefly tell us about this presentation for the audience, because audience are pretty.
Sorry, I can't hear you clearly, but are you asking me to explain a little bit about his lecture?
Yeah, if possible, that would be great, because audience are sitting and they're just expecting something from this lecture, then we'll start the next lecture on time.
Okay, he's actually explaining TCM, which is a formulation by a medicinal plant. This formulation is designed by him. He's basically a medical TCM doctor. He has used this formulation to treat the blood problem in China in his hospital. He's the director of the hospital, and this TCM is used for the many years for the patients who came to this hospital. So basically, he's introducing this TCM.
Thank you. What about this particularly his TCM as far as I understand the traditional Chinese medicine? So he is particularly talking about particular one medicine, or he is talking about the general medicine? Hello, Jan. Jan, your microphone is on silent. Jan, we can't hear you. Your microphone is silent. Hello, Dr. Dong. Are you available to start your long talk?
Sorry, I just called Professor Yang to confirm. I think that杨院长，您能听见我们吗？杨院长，我们看不到您，杨院长，现在是卡住的，听不到也看不到。问题是我们看不到您，也听不到您那边，您那边是没有声音也没有影像的。咱们就继续下一个吧，因为这边完全听不到也看不到，可能没有办法再继续了，他们已经着急了。你看行吗？
Dr. Yan, can we start the second talk? Meanwhile, if the problem is resolved, we can resume the first talk once again. Is it possible?
Dr. Zahi, I can't hear you clearly. I'm not sure what you say. So I think we should call next because I also not able to listen that side.
Okay.
We should continue next. It's take too much time.
So next speaker is available. We can start right now.
Professor Liang, are you there?
OK， 请见。OK。
Okay, then we will start and move to the second speaker of the talk, the second session, the Dr. Professor Dong Liang. He is basically his expertise in the pharmacology and biotechnology. His talk is anti-neuroinflammatory ingredients from the ethnic medicinal plants of Papaya and Sapien genus. So, mic is over to you, sir, please.
老师，您听得见吗？ 老师您听得见不啊？
他可以有。
Professor Leon, are you there?
Yes, I'm here. I think we should stop the first one, but the first one is still ready to continue. Oh my God.
Okay, do it. But
喂，
哎，王老师，王老师，您能听见我们讲话吗？
王老师，
听见看不见，不是什么原因？
哎，杨院长，现在可以了。
对啦，对吧？
现在可以听到。
对吧？看得见不？看得见不？
看得见，看得见。
那下面我们继续啊。花一点点时间，我们花一点点时间啊。听得见不？喂，
可以听得见，杨院长。
好，那我就继续啊，各位啊。好了，我们研究内容期啊，智能合同胶囊通过 NLRP3 炎症小体抗动脉粥样硬化的机制研究，研究结论啊发现，智能合同胶囊能够改善高脂血症、颈动脉粥样硬化、加速血脂及减少颈动脉斑块生成的作用。研究方向把智能合同胶囊抑制 NLRP3 炎症小体合化抗 U937巨噬细胞交往的机制研究。那么研究结论发现，智能合同胶囊能够通过 NLRP3 信号途径抑制 U937巨噬细胞交往的作用。研究方向9，基于内质网应激探讨智能合同胶囊这个对 OXLDL 诱导巨噬细胞凋亡及泡沫细胞形成的影响。结论发现，智能合同胶囊能够有效改善 OXLDL 诱导巨噬细胞凋亡及泡沫细胞形成，可能与 SIRT7 介道的内质网应激有关。研究内容是基于脂质组学说的智能合同胶囊抗动脉粥样硬化作用机制研究。研究结果发现，智能合同胶囊可以降低血脂及改善动脉粥样硬化，其主要作用通路为甘油磷脂代谢及甘油脂代谢。研究内容是智能合同胶囊对气血型老年痴呆模型大数的干预研究。研究结论发现，智能合同胶囊能够有效改善老年痴呆模型大数认知学行为的作用。那么前述啊，仅列了部分的围绕智能合同胶囊开展的基础研究，涵盖了脑出血、脑缺血、动脉粥样硬化、高血脂、老年痴呆、炎症与免疫等等。那么它的主要机制有调节血脂、调节炎症因子释放、抑制交往、改善血脑屏障有关系。那么从临床研究来看，我们做了一些研究。第一个研究呢，对帕金森病的临床研究，研究结果发现，智能合同胶囊能够提高帕金森病患者的临床疗效，有效的改善患者症状，提高生活质量，无明显的不良反应和在临床推广应用。第二个研究内容就是对颈动脉粥样硬化的患者的临床疗效的观察，发现智能合同胶囊具有抗动脉粥样硬化和稳定斑块的作用。研究内容3，智能合同胶囊对缺陷脑小血管的并合并血管性认知功能障碍的临床疗效的观察，发现它能够 改善缺血性脑小血管病合并血管认知功能障碍患者的日常生活自理能力及提高认知的功能。研究内容是智能合成中胶囊对缺血性脑卒中患者急性期的脑水肿及 HSCRP 的影响。那么结果发现，智能合成中胶囊可以减轻脑水肿的形成和发展，改善神经功能损伤，对缺血性脑卒中急性期有脑保护的作用。研究5，智能合成中胶囊结合中医辨证治疗缺血性中风急性期30例临床观察发现，在辨证认知基础上配合使用智能合成中胶囊，能够明显提高缺血性中风急性期患者的临床疗效，在改善神经功能损伤方面具有积极的意义。研究内容6，智能合成中胶囊对风痰瘀阻型急性缺血性脑卒中患者外周血的这个影响细胞因子及神经功能的影响，发现可以减轻炎症反应，保护脑细胞，改善患者的预后。研究方向7，智能合成胶囊对心血管疾病患者血脂影响的 meta 分析，结果发现能够改善心脑血管疾病患者的血脂，而且没有明显的不良反应。研究方向8，智能合成胶囊对老年高血压患者怀壁指数及高敏 C 反应蛋白同型半胱氨酸的影响，结果发现智能合成胶囊能够改善其动脉硬化的程度。那么我们在一年的科研项目就包括智能合成中胶囊加麝香花与可丽，治疗脑出血的多中心垂直双盲安慰剂对照研究。这个研究呢，我们在2021年1月1号项目首例病例入组，截止目前呢，我们项目已经入组了病例共计229例。那么智能合成中胶囊在临床上治疗疾病有效率总有效率有明显的提高，包括脑出血这个后循环缺血、脑缺血、帕金森病。总之智能合成中胶囊开展的临床研究包括急性脑梗死、脑出血、帕金森症、高血压、血管性痴呆等诸多研究。上述相关的临床研究均表明该药能够提高临床的疗效，改善预后，值得临床推广应用。目前呢，我们取得了一些成果，这个是我们智能合成中胶囊历年发表文章的这个趋势图相关的文章大约10多篇。2011年5月，我们获得了中华人民共和国国家知识产权局发明的专利。那么自2006年起，我们在医院使用了5万多瓶，每月产值达到了22万这是我们今年的10多年的智能合成中胶囊的用量的统计逐年增加。那么这个是我们收入的统计，也是逐年的增加。目前呢，智能合成中胶囊推广于川滇黔渝150多家医疗单位，年均治愈及好转率达到了10%到20%提高临床疗效达10%到20%，减少发病率、致产率及死亡率达5%到20%。今年8月，我们智能胶囊也成功纳入了四川省医疗报销目录，而且我们建立了川南悬浮学术流派工作室，相关的研究获得国际科研立项资助20多项，而且获得了国家自然科学基金项目2项。2018年，我们获得了第一批省科技厅科技计划支撑项目。2016年6月，我们智能胶囊防治动脉粥样硬化关键技术研究科技成果评价达到了国内领先水平。我们先后获得了科技进步奖10余项，包括中华医学会3等奖、中华中药学会3等奖、华夏医学3等奖。这个华夏医学这个科技奖认为我们有5个创新点，一个悬浮理论新体系， 2. 率先运用风药开选防治中风。3. 率先运用风药开选防治血管性痴呆。率先运用风药开选防治头痛。率先运用风药开选防治眩晕病。我们而且获得了中国中药促进会科技进步2等奖、3等奖，四川省科技进步3等奖2次，四川省科技中药学会科技进步2等奖1次，泸州市科技进步1等奖2次。参与编辑了这个专家共识有2项，近5年参编各级各类教材有5部，主编参编专著20余部。这是我们出版的部分的专著团队建设方面，我们拥有数十项国家级荣誉称号，包括国家高级促进中心、国家临床重点学科及专科。团队建设方面，包括国家区域中医诊疗中心1个，获批国家高级促进中心，2批国家中医临床研究基地、心源性注重防治基地、国家中药服务出口基地、四川省临床医学研究中心、国家中医心血管病临床医学研究中心分中心、国家中药传承中心团队为会员立场单位牵头成立中国中药信息研究会老病分会，牵头成立了国家级学会百求恩精神研究会中医心脏病学专委会。以上呢就是我简要给大家介绍了我们这个院内纯中药制剂智能回农胶囊的实验研究、临床研究以及取得一些成果，不当之处希望大家批评指正。Thank you for your attention， 谢谢。找一下会儿。
Priyan, can you on behalf of us can you appreciate it? Dr. Yan,
Hello.
can you please appreciate him on behalf of ISNPC?
This is a medicine. Basically talking about this already treat for the cardiovascular disease for about 2 million people in his hospital and the about 150 hospitals in the south of China. So this is a very successful TCM which already won lots of awards as well. And he is a director of the hospital, so he also try to probably in the future can do the clinical trial in Pakistan and register in Pakistan as well. 杨院长，我们就到下一个了哈。谢谢您。
谢谢啊，好。
So Dr. Zahid, we can go to next.
这是王艳。
王艳啊。
您那个关一下。
好，关一下。
杨院长那边麻烦您关一下话筒。
已经关了。
Now the second, I invite the second speaker, Professor Dong. Professor Dong, can you hear us?
Professor Liang, can you hear?
Yes， 可以。
Please go ahead.
.能看到吗？
Yes, yes.
可以了啊，开始了啊。Thanks. Hello. Thanks for giving me a chance to speak here. I'm Liang Dong from the State Laboratory for Chemistry and Molecular Engineering of Medicinal Resources, Guangxi Normal University, located in China, Guilin, a very beautiful city. Today, I want to introduce our work, study on the anti-neuroinflammatory components from ethnic plants of the genus Piper and Sepium. I will present
放大一下
my-
PPT， 放大一下 PPT。
I guess your slide is not sharing with us properly. We cannot see your slide mode, or did you change it?
放大一下，可以吗？这个
放映
看不到吗
放映？PPT， 现在不是放映模式。
咋放映？
他们说您声音有一点小，
那我声音弄大一点吧。
离话筒近一点。
这个我是放映呢。
啊
共享。
没有
啊
放映，现在点从头开始就是放映了吗？是共享的，但是那个偏 PPT 不是放映模式？
我看一下啊。现在可以吧？
没有，
现在呢？
没有看不到。您是把这个 PPT 打开以后再分享 PPT？
先把 PPT 打开是吧？
对。
现在可以了吗？
可以看到 PPT， 但它不是那个放映模式，就是可以编辑的这种模式，没有放映出来，就可以看到左边这好多的页面都在那，1、2、3、4、5、6。
那
对，从头对，从头开始。
是先打开吗？从头开始，现在可以吗？
啊，对，点一下。
现在可以吗？
还没呢。放映不了是吗？那要不就这么讲吧。
我这儿能放映呢，是先打开这个还是后打开？
是要分享屏幕的时候要分享这个 PPT， 然后就可以放映了吗？
共享屏幕是吧？
对，选择幻灯片那个界面。
哪一个？是幻灯片放映吗？
就是你点了放映了，但是我们看不到放映模式是吧？那要不就这么讲吧，反正也能看见，就稍微小一点点。
现在可以吗？
没有，我们看到的不是放映模式。
那咋办？
那就这么讲吧，就是稍微小一点点那个。
那我把前面左边的给它关着吧，这个关着，这样可以吗？
那应该好一点吧。最大化呢，把这个窗口最大化。
As long as we can hear you, I think you can start your talk.
这样呢？这样可以吗？
看不清楚底下那个部分，只能看到上一半，没有放映出来。
这样呢？
现在可以看出整个的，就这么继续吧，我觉得这样是可以看见的。
这样呢？
没有变化。
能看得清楚不？
看得清楚。
那我就这么讲吧，我也不知道问题出哪儿了。那我就从头开始讲吧。Today I would like to introduce our work study on the anti-neuroinflammatory components from the ethnic plants of the genus Piper and Sepium。现在声音怎么样啊？
可以。
I will present our work from the following 3 sections. First, about background. As we all know, located in southwest of China, Guangxi is rich in medicine resources. According to the newest report, there are about 7,088 kinds of traditional Chinese medicine resources in Guangxi, including 5,996 kinds of medicine plants, and a lot of medicine animals and marine medicine organisms, which ranked first in China. Many famous medicines, such as in Chinese name Lo Hanguo and Ji Xue Ten, were mainly distributed in Guangxi. Also, Guangxi is rich in ethnic medicine resources because of a large population of ethnic minorities such as Zhuang, Yao, and Anak live in Guangxi. Up to now, there are 2,285 kinds of Zhuang medicine resources are recorded in list of Chinese Zhuang medicine resources, and more than 970 kinds of Yao medicine resources are included in Yao ethnic medicines in China. Our laboratory have been focused on the research on Yao medicine, which is recorded in Yao ethnic medicine in China. In this
师兄，你是
book,
换页了吗？我们只看到了第一页，到现在还是第一页。
是啊，我换页了。
我们只看到的啊，现在看到了。
不是，我现在退出来了。
啊，
非得要退出来你们才能看得着吗？
有可能。现在看到了，刚才就一直是封面页。
是不是很小啊？
还可以，继续吧。
那我先看一看吧。哎呀，怎么回事？不好意思啊。I'm very sorry. 我从这儿开始讲吧。First, about the background. As we all know, located in southwest of China, Guangxi is rich in medicine resources. According to the newest report, there are about 7,088 kinds of traditional Chinese medicine resources in Guangxi, including 5,996 kinds of medicinal plants and a lot of medicinal animals and marine medicinal organisms, which ranked first in China. Also, Guangxi is rich in ethnic medicine resources. Up to now, there are about 2,285 kinds of Zhuang medicine resources are recorded in list of Chinese Zhuang medicine resources, and more than 970 kinds of Yao medicine resources are included in Yao ethnic medicine in China. Our laboratory have been focused on the research of Yao medicine, which is recorded in Yao ethnic medicine in China. This book, for the recent years, the focus was the genus paper and the spectrum. The genus paper belongs to the family Papyraceae with about 2,000 plants worldwide, such as Papacy type Papyrus, and the genus Spectrum belongs to the family Euphorbiaceae with about 120 species worldwide, such as Spectrum discarnate and Spectrum sebriform. From the chemical point of view, the genus paper are rich in neuronigrans and alkaloid. This is the structure of neuronigrans in the genus paper. This is alkaloid, upward alkaloid. And the genus Spectrum are rich in coumarin and terpenoid. This is the structure of a coumarin derivative. This is a diterpenoid. This is a tritopenoid. The above structure all interested us. From the bioactivity perspective of view, our laboratory have been focused on the neuroinflammation. Neuroinflammation is involved in the pathogenesis of a variety of neurological diseases, including neurodegenerative diseases such as AD, PD, as well as other neurological diseases such as ischemic stroke and depression, and microglia are inherent immune effector cells of the central nervous system. The activation of microglia has become a hallmark of neuroinflammation in the brain. With the development of research, it was found that neuroinflammation mediated by overactivated microglia is one of the key targets in the prevention and treatment of AD and other neurological diseases. And modern pharmacological studies have shown that the genus paper and sepium have anti-inflammatory, antioxidant, and protective effects on the nervous system, which can be associated with neuroinflammation. Our laboratory have found a series of L drugs with significant activity of inflammation in the anal stage. So whether there are active lateral products in the genus sepium and paper, that's a focus of our research recently. The representative works could be divided into two parts: the genus paper and the genus sepium. Section A-23 about the genus paper, including 2 paper plants, and the section D-2G about the genus sepium, including 4 sepium plants. First, the first part is the genus paper. Section A about study on the anti-neuroinflammatory cycloneuronicotinoids from Piper Hansen. Piper Hansen, its Yao medicine name is Xiao Changfeng, and its root and stem could be used for the treatment of rheumatism, wound cold, and inflammation, and so on. We started to investigate this plant from the year 2015 and found a series of nicotinoids which connected by carbon-8, carbon-3, or carbon-8, carbon-1. In the biological activity test, we found that the nicotinoids connected by carbon-8, carbon-3, the above type, their activity is better than the below structures. For the most potent active compound 1, they carried out in-depth research and found the compound 1 could inhibit the inflammatory factor production by inhibiting TLR4 NF-κB signaling pathway in LPS stimulate BV2 microglia cells. So whether because of a large amount of neonicotinoid have been found in the genus paper. So whether there are other neonicotinoid with a richer amount and or normal screen, is our research. In the continued research, 2 pairs of cycloneuronicotinoids in enantiomers and their Piper Hansen A and Piper Hansen B and their biosynthetic precursor, 2 pairs was also found. For the cycloneuronicotinoids, they were made up by 2 6 carbons, 3 carbon moiety through 1 coupled through 1 ring or more rings. They often exist in pair in nature, so they measured their optic and carried out chiral separation and single case derivation data analysis and confirmed their existing in pair. Their absolute configurations was confirmed by experiment and calculated ECD spectra. For the 2 pairs of cycloneuronicotinoids, Their structure is there in the nature. The cyclic neurons up to now could be divided into 10 subtypes, of which 8 subtypes was covered through the first 2 8s were covered through 1 gene, and the 9th subtype were covered through 2 genes, and the 10th subtypes were covered through 3 genes, and Piper-Hansen B compound 2 was consistent with the 9th subtype, and our isolated compound 1 Piper-Hansen A represented on unprecedented carbon skeleton. We deduced the process biosynthetic pathway of compound 1 and 2 and successfully verified the form on known compound. In the biological activity assay, we found that different enantiomers have different activity. Although compound 2 has the most potent activity, because compound 1 has a novel skeleton, we choose this compound for in-depth research and found it could inhibit the mRNA levels of pro-inflammatory cytokines in BV2 microglia cells, which suggests that it has the potential to treat inflammation. Next is about anti-neuroinflammatory alkaloid from piperpopernum. Piperpopernum, its young medicine name is dubindan, and its whole plant could be used for the treatment of blood circulation, pain, and rheumatic disease. In our study, 23 compounds were isolated. The red marked amino, including compound 1, 2, 6, are new tyrosine type alkaloid, and compound 7 to 11 are alpha-fine alkaloid with different ratios of positional isomers. The orthogonal configurations of 7A, 7B, and 810 was confirmed by XZ diffraction date. In the anti-neuroinflammatory activity test, we found that the red marked compound 10 to 13, they had significant inhibitor activity. This is their activity. But for the same alpha-fine alkaloid, compound 7 to 9, they were almost have no effect, which suggested that the alpha-fine alkaloid with this unit, methylene dioxide, is a key pharmacophore. The same phenomenon was existed in compound 15 to 17. Compound 17 had a good activity, but compound 15 and 16 were inactive. They all suggested that the methylene dioxide is a key pharmacophore for the anti-neuroinflammatory activity of alpha-fine alkaloid. Last is about the anti-neuroinflammatory neonicotinoid from piperpopernum, the same plant. What interests us is that the neonicotinoids exist in this plant also existed in pair, and they had a zero connection model. Compound 1 connected the two propanoid were connected through carbon 2, carbon 9, and compound 2 were connected through carbon 5 and carbon 6. And the orthogonal configurations of compound 1 was confirmed by single crystal XZ diffraction date, and compound 2 Its operational configurations was confirmed by experiment and calculated ECG spectra. In the activity test, we found that their activity is weaker than the above alkaloid, and compound 1A and 1B had different inhibition, and we did in further condensation confirm that compound 1 could inhibit mRNA levels of anodes and other pro-inflammatory cytokines, including interleukin-1 beta, interleukin-6, and TNF-alpha in LPS-induced BV2 microglial cells. Next part is about the genus Sepium. The genus Sepium belongs to the family Euphorbia, which had been paid attention for many years by the scholars of natural product chemistry. First, in this part is about the anti-neuroinflammatory coumarin and tacrocin tritopenoid from Sepium discana. This plant, its young medicine name is Day-02, and its stems and leaves in ethnic minorities could be used to treat snake bites, allergic dermatitis, and eczema. And a series of coumarin lignoid and tacrocin tritopenoid was isolated. For this type of natural product, tacrocin tritopenoid, they were relatively rare in nature. Up to now, only 4 tacrocin tritopenoid was isolated was found in the genus Sepium. This is the first report of tacrocins in the plant Sepium discana. For the coumarin lignoid, they will often exist in pair and often angularly fused, but the coumarin lignoid in our research were all linearly fused, and we confirm their existing in pair through chiral separation, and also confirm their operational configurations. For their anti-neuroinflammatory activity test, we found that the red marked compound 1 to 9 had good activity, but compound 4 and compound 10 to 14 were most inactive, which suggests these 5 compounds all have these 3 moiety, which suggested that the introduction of these 3 units is critical for their anti-neuroinflammatory activity. Section E about the anti-neuroinflammatory diterpenoid and tritopenoid from Sepium rotenfolium. Its air part could be used for the treatment of eczema, scabies, and detoxification. It was often found in coastal areas of northwest Guangxi, and a series of diterpenoid and tritopenoid was isolated. The red market was 7 new compounds, including 4 new diterpenoid and compound 12 to 14 were new tritopenoid. These 4 new diterpenoid, their operational configuration was confirmed by X-ray induced ECD spectra and experiment ECD spectra. For all the new tryptopanoid isolated in our study, their observational configurations was confirmed by X-ray diffraction data analysis. And in the activity test, we found that compound 3, compound 8, and compound 11 showed excellent activity, which suggested that terpenoid was an active component for in the plant's sepium return dephonium. Last is about the antioxidant chapunic acid derivative from sepium bacterium. From this plant, a serial chapunic acid derivative was isolated, the red marked anneal. In this study, compound 6 to 10 had a 0.011S configuration. Their configuration, including the negative and opposite configuration, was confirmed by Newman projection, single crystal X-ray, and experiment ECD. Although they were all inactive in the anti-inflammation test, but they showed prominent DPPH test showed mini antioxidant activity and we chose compound 1 to do further study and found this compound can inhibit the mRNA levels of antioxidant enzymes in Day-02 microglial cells. The last part is about the anti-neuroinflammatory phenomenal glycoside and phenylpropanoid glycoside from Sepium japonicum. This plant, its stems and leaves could be used for the treatment of eczema cases detoxification, and 15 compounds, including 5 new compounds, 5 new flammable glycoside and a new phenylpropanoid glycoside were isolated. In our activity test, we found that compound 1, compound 8, and compound 9 showed excellent activity, and compound 7 and compound 10 showed weaker activity. We focused on compound 1 and compound 7. They were showed potent activity with the galactose containing fragment, but compound 11 and compound 12 had the galactose containing fragment was almost inactive. So the perimetry structure activity relationship is that the anti-inflammatory activity of galactose containing fragment in flavonoid glycoside was significantly better than that of galactose containing fragment. The next part is about our summary and thoughts. Our research have further enriched the structure types of neuronigrans in the genus paper, and in this paper is rich in neuronigrans and alkaloid. They will also be our focus in the future. For the function of promoting blood circulation and removing blood stasis, the research of the genus paper will be launched around the model of ischemic stroke nature, and we have found a small molecular alkaloid and a study on it. For the genus Sepium, a series of casein diterpenes were isolated. Among the 5 separation plants we isolated, 3 of them exist this type of diterpenoid. So from the I say husband diterpenes could be used as a marker of the genus Sapium. And from the perspective of chemical taxonomy, our work verified that it's scientific and reasonable to correct the Latin name of Sapium. And in our study, we also found that husband diterpenes showed potent anti-inflammatory activity. And we are carried out in-depth study on one of them. From the perspective of topic selection, we are only studying the chemical components of the ethnic medicine plants. So in the future, we want to deeply excavate the nature active component of ethnic medicines and in nature under guidance of modern scientific theory and in compilation with focal unilateral prescription, tested prescription, and focal transient uses. The last is the representative paper on the genus paper and Sapium, including 1 paper in Journal of Organic Chemistry and 2 papers in Journal of Nature Products, 3 papers in Bioorganic Chemistry. That's all. Thank you.
Thank you, Dr. Dan. Unfortunately, we are short of time, but I really appreciate for your wonderful presentation and a lot of results you shared with us. I really appreciate for your time and thanks for your presentation. Now I will invite the next speaker. The next speaker of the session is Professor Dr. Shabana Sindhi. She is one of the most prominent scientists of Pakistan in the field of neurological disease, and she is going to share with us her live doctor research with us. Dr. Shabana.
Okay, thank you.
Thank you.
I think I still have some time left in my life. Okay, here I'm going to share part of the research data from my study in the field of the neurogenesis. Can I proceed now? There seems to be some trouble in this. Can I proceed now? This is for the forward end. Thank you very much. Okay, so I was saying that I'm going to share part of the data from my study in the field of the neurogenesis. And we all know that basically brain is a very complex organ. It consists of millions of cells, billions of cells, which are in continuous communication with each other, right? And this communication within the cells is beautifully maintained by the harmonized play of the chemical or electrical impulses in the central nervous system. So in case if this communication, any insult to the brain, which affect this communication can easily lead to the sequences of the events that will going to affect the overall function of the brain, and that also could lead to the neurological disorders. And neurodegeneration is one of the most common neurological disorders that affect almost around 1 million of individuals worldwide. And basically, what happens is that like I said, that it affects the various individuals worldwide. And in order to, despite the fact that day by day our understanding about the brain is advancing, but yet it is still the area where we lack appropriate understanding of coming up with a treatment which could either halt the further generations of the could halt the further degeneration going on in the brain, or else at least start replacing the already degenerative regions in the brain. So basically, this neurogenesis is a big question of that exists in the neuroscience, that whether it is possible to initiate the process of neurogenesis in the adult brain or not, because this is a well understood fact that the neurogenesis occurs in the neonatal brain, but it was a dogma in earlier than 1950s that once the brain develops, the further regeneration potential of the nerve cells halts. But in 1950s, then the literature started to report that even in the adult human brain, there is a possibility of initiating the population of the cells which resides in specific regions of the brain, which can be initiated and can be destined to become mature neurons. And basically, this is what is called neurogenesis. Like I said, that earlier than 1950, it was a belief that no new neurons can be generated. But later on, it was shown that new neurons can be generated in the adult brain, and it was documented with the proof, especially in the animal models. However, it took around 30 years to show that even the human adult brain has the potential of generating the new neurons, depending upon if we give a specific or activate specific endogenous modulators or regulators which are present in the CNS. If we can initiate those regulators, endogenous regulators, then it is possible to initiate this process of regeneration and reversing back the damaged area by recruiting the cells into the specific niches of the brain where the damage is already there. Apart from the endogenous modulator, there are some extrinsic factors as well, which I'm going to show you in other slides, which can be used. For example, there are natural products, special natural products that also can have the ability to initiate the residing populations of the immature NPCs, which are neuroprogenitor cells in the brain, can initiate them and destine them to become either into the neuronal lineage or into the glial lineage, depending upon what is the need of the nervous system at the time of the insult, right? So it can go both into the glial lineage as well as in the neuronal lineage. Now the question arises that if it is possible to initiate the neurogenesis, is it a random event which can occur throughout the brain, or are there specific regions where we have the residing neural precursor cells which can be initiated and further destined to become the neurons or non-neuronal cells. So basically, to answer that question, we have the specific regions in the brain where the populations of the so we have these specific regions in the brain where we have a large populations of the cells which have the potential of becoming mature cells, neuronal cells at the time of requirement. And these regions are basically in the subgranular ventricle region, which is near to the ventricle of the brain, as well as in the subgranular zone, which is in the hippocampal region. So these two regions are the specific region where you have abundance of the NPCs, right? Okay. We have to keep in our mind that neurogenesis is not the process that will just go into that in a startup or initiate in a very random fashion. It can be initiated only at the time of the need. Continuous process of the neurogenesis, example of the continuous process of the neurogenesis, we can see in case of the memory development, right? And this memory development is associated with the hippocampus. So that is one of the reasons that why the nature itself has kept this population of specific NPCs in that region, which is associated with the development of continuous development of learning and memory, right? So here there's a large population of residing NPCs present. So in this region, there are certain regulators which can regulate or initiate this event, which can destine the cell to become the specific fate into the neuronal or non-neuronal lineages. These regulators are either, there are certain neurotrophic growth factors. We already have this endogenously present neurotrophic growth factors such as nerve growth factors. Then there are certain other factors like brain-derived neurotrophic factors. These factors are basically responsible for the growth of the or regeneration of the nerve cells, right? Apart from this neurotrophic growth factors, sometimes extrinsic growth factors are also incorporated as far as the experimental studies are concerned, right? Besides this neurotrophic growth factors, physical activity like exercises, that also initiate this process of neurogenesis. Stress, it is a regulator, negative regulator. So there are certain positive regulator as well as the negative regulator of the neurogenesis. So stress plays a very detrimental role in the neurodegeneration in such a way that it is a negative regulator of the process. Aging is also one of the negative regulator of this process. Inflammation is kind of a very disputed factor, because some of the studies have shown that inflammation initially activate the neurogenesis, but at up to a certain level, and then there are certain other regulators secreted by the inflammatory cells, which then act as a negative regulator. So inflammation has been reported in the literature as the negative as well as the positive regulator of this process. Apart from this, there are certain neurotransmitters like dopamine, serotonin. They are also positive regulators of the process of neurogenesis. So these are basically the regulators, either negative or the positive regulators of this whole sequences of event going on in the CNS. Okay. So basically, why nature has kept this process in our system, a process of neurogenesis. But now we know that it is now accepted fact, which used to be a dogma that it cannot be occurred in the adult brain. It is now not accepted, it is rejected. It is possible in the adult human brain as well. And the best example, like I told you in the earlier slide, that it is well documented in case of the development of memory and learning. And learning and memory development is a lifelong process, right? Continuously taking place on a daily basis, from minute to second to milliseconds, right? So this is the well documented process in case of the memory. So neural plasticity. This is the mechanism which is involved in the memory development, right? So it is playing a very important role, the adult neurogenesis, in the neuroplasticity, and specifically in case of the hippocampus, right? Then adult neurogenesis also plays a very important role in keeping the homeostasis in the central nervous system and maintenance of this homeostasis system throughout the CNS. There's some factors in preserving the cognitive functions and repair of the damaged brain cells. They are always affected by the brain disorder. So seeing the roles of this neurogenesis in common physiological function of this neurogenesis, which is going on in the adult human body. I also developed my interest at this stage because as a neuropharmacologist myself, I think that if it is possible for us to show that there are certain extrinsic factors specifically present in the natural products, because our institute excels in the chemistry of the natural products. So I also try to restrict myself to the use of the natural products as well. So if we have any extrinsic factors present in the natural products, and if we can halt the further degenerations of the nerves, and also able to revert back the damages which are already caused over there, so then it might give a hope to the patients who are suffering from debilitating neurological disorders associated with the degeneration, right? This slide is showing you the clinical conditions which are affecting the neurogenesis. Like I said, that they are just yeah, exactly. So stress in case of a depression, irradiation, inflammation, meningitis in case of the infections, right? And also in case of the chronic alcoholism. These are all the negative regulator of the neurogenesis, which decreases the potential of the residing NPCs to participate fully in the CNS system. And you know that even at the time of the stress or inflammation or infections or alcoholism, your memory and learning capacity or potential is also compromised, right? That's simply because they are the negative regulator. As far as the positive regulator are concerned, you can see over there ischemia, seizures, trauma, and dementia. This is very surprising to see these factors as a positive regulator. But let me tell you that in case of the seizures, in case of the trauma, in case of ischemia, the very first event that I initiated is because the body is calling for the help, just like the immune system, right? When you are suffering from the infection, what happens? Your immunology responses are initiated and they recruit the inflammatory cells over there. So in the same way, when ischemia occurs, seizures occurs, trauma occurs, the very first role of your CNS is to recruit the cells which can halt this event. So in that cases, up to a certain level of the regeneration of the nerve cells or neural progenitor cells are documented in this case. But after a certain stage, then this process starts declining, right? So then again, you have to be very careful. Even in the case of the cancer, there's a neurogenesis. I'm talking about the CNS cancers, right? Tumors. Over there, there's a exaggerated generations going on. So we have to be very careful that how we want to address the question or how we want to address our study target, right? Okay, clinical applications of NPCs are in the cerebral ischemic injury, in the, like I said, neurodegenerative diseases, multiple sclerosis, brain tumors, and congenital malformations. These are some of the area where clinical applications of neuroprogenitor cells are widely studied and are now started documented in the literature as well. Again, there are specific. Now, if you want to study neurogenesis, you have to keep in your mind that it's not. It is like I said earlier. It is not a random mechanism to follow, right? So in that case, you need to know what are the stage-specific markers. When I say stage-specific markers, means the immature cells need to first proliferate, then they need to differentiate into a specific lineage of the cells, and then they are destined to incorporate it in the area where they are required. Right? So you need to know the stage-specific markers in order to follow your study. Right? So these are certain markers which are shown over here, which shows that whether this, if you get this GFAP, vimentin. So what does it specify? It specified the, like for example, nascent, it specified the immature neuronal lineage, right? GFAP, glial fibrillary protein, that specified the glial lineage of these cells, and so on. And in the case, such is the case with the mature and immature neurons as well. If you get this markers, specific cell-specific markers, then it shows you that the cells you are getting in your study, they are either immature or mature neurons, right? So in order to study, or in order to know what are your experimental data are showing, you need to know very first thing that which marker you are going to study, and it depends on which lineage of cells you are targeting. Okay, this is showing some for other markers as well. Okay, apart from this cell-specific markers, there are signaling mechanisms which are involved in the neurogenesis, and these are the signaling pathways which can be studied or which can be chased in order to see whether your target question can be answered in the frame of the signaling pathway, right? Okay, like I said earlier, that by seeing all this, going to the literature and seeing the role of the neurogenesis, how it can play a role in the regeneration and reversing back of the damages, then I developed the interest in this. And for that, initially I have focused, we focus our study on the use of the natural product. Natural products are various plant-derived natural compounds, and many of them are reported already in the literature to be efficacious in promoting the differentiations of the neuroprogenitor cells. These natural compounds have the potential on neuroprotections, and neuroplasticity are the great interest to the neuroscientists. Okay, keeping this in mind, we started our study with, okay, before going into the compound, I'm just going to briefly show you the methodology that we have used in our study. We have prepared our own primary cultures from the rat pups, 1 or 2 days old rat pups. We have dissected out the hippocampus and cortex from the pup brain, and then we have cultured in the dishes, right? Once we get the confluent culture of the primary cells, then what we have done, we have plated them in a 96 well plate. And very first thing that you can see after getting the hippocampal and cortical cells, right? Very first thing you need to show that whether your compounds have the potential to proliferate the hippocampal or cortical cell or any other cells that's harvested from the brain, right? Depends upon what question you are going to answer. So in our case, we have performed, after having the conference primary cultures of cortex and hippocampus, we have tested various compounds from the natural product through MTT assay. So basically, MTT assay gives you only the information that your cells are proliferating or not as a result of the compound that you are testing. Right? So what we have seen that after getting the proliferation in our study, we move on to the LMR and Burdue assay. Burdue assay is basically shows you whether your cells are rapidly dividing or not, right? Once we get this results in the MTT assay, then we move on to the analysis of the gene expression of the markers that we are interested in, right? And then we also did the immunocytochemistry. So basically in MTT assay, we have screened various compounds. I'm not going to go into the principles of MTT assay or Burdue assay. Just going to go quickly through the slides of the results. So we have tested various compounds, but this 4 compounds have given us the maximum proliferative activity, right, in the MTT assay. But I'm going to focus my results on this easy isoxazolone. That's simply because this is the US patented compound of Professor Dr. Taur Rahman and his research group, including Professor Dr. E-Coli and Professor Dr. Farzana Shahin. So we started with this compound because it was already patented as an anticonvulsant compound, and we wanted to see whether this anticonvulsant compound can also initiate the neurogenesis to repair the damaged area. These are the patent, and last year we got the research group also got the patent on the neurogenesis potential of this compound isoxazolone. So this is just showing you a phase contrast microscopy of the MTT assay of the corticals and the hippocampal cells, and you can see after 24 hours how these cells are proliferating. But in case of the hippocampus, you can see over here that it's not only proliferating, but it is also differentiating, right? This is for the hippocampal cell. Earlier one was for the cortical cells. So you can see here, after 24 hours and 48 hours, not only the cells are doubling, but they are also differentiating in case of the hippocampal and cortical cells. But this pattern you cannot see in the control cells with having no other treatment. Then we move on to the Burdue and LMR blue assay just to see whether they are dividing cells or not. And what we have observed is that the treatment of the isoxazolone in case of hippocampus as well as in the cortex, they significantly increases the actively dividing cells. So then once it was proved that our compound is active enough to support the growth, proliferation and differentiation, so we move on to the see whether it can also helpful in hypoxic condition or not, because we have to keep in our mind that ischemic stroke is one of the conditions which most of the people are suffering around the world, and that also produces a lot of damage in the brain. So we wanted to see whether this compound which is having a potential of the halting this degeneration, is possible to stop the hypoxic damages as well. So what we have observed that the treatment of the compound in case of the cortical cells, you can see again that it has significantly increases the number of the cells, proliferating cells, which are actively participating in multiplication and differentiation. Same results we have got for the hippocampus as well, right? So in both cases, cortical and hippocampal cells, these are the region where we can reverse back the ischemic damages if appropriately treated with the isoxazolone compound. Then we have, like I said, that we were interested to see whether this compound also support the differentiation or not, and for that we have done the immunocytochemistry. For the neuronal lineage marker, we have used NASTIL. Then we have used the glial progenitor marker. Tubulin and claritinin. The tubulin is immature and calritinin is the marker for the mature neurons. So in case of Nastin, you can see over here that the isozylitol treatment not only increases the Nastin immunofluorescence in the hippocampal cells, but it also increases in the cortical cells as well. This is basically the cortical cells, and you can see the staining of the Nastin over here. This is just now showing you quickly a graphical representation that both in hippocampus and cortical cells as compared to the control cells, you can see how significantly it is increasing the staining fluorescence of this Nastin, which shows that the Nastin containing population of the cells are doubling, right? Okay, then we move on to the GFAP. We have also seen the GFAP in the cortical region, but in case of the hippocampal cells, although we have seen the GFAP, but it wasn't as significant as we observed it in the cortical region. Okay, then we move on to the tubulin, which is the marker of the immature neurons. And again, over here, we have seen that in cortex, we have observed the presence of the tubulin, but as compared to the cortical cells, we have seen that in hippocampus, the immunofluorescence of the immunofluorescence was more as compared to the cortical region. It means there are more population of the immature neurons as compared to the cortical regions. And then move on to the claritinin, which is the mature markers. Again, we have seen that in case of the hippocampus, we have seen significant increase over here in the mature neuronal markers in case of the hippocampus. I'm not going to go into the detail of the genetic expression, but just quickly I'm just going to give you the conclusion. We have also done the transcription factor analysis and seen the same results as compared to the other markers. So concluding the talk, the isozylitol have growth supporting activity on both hippocampus and cortex progenitor cells, which may be a part related to its ability of proliferation in normal and hypoxic conditions. And activity might be related to the ability of isozylitol to modulate the expression of underlying molecular markers. And in future, we are going to try to make the animal model of neurodegeneration and to see if this compounds can show the same or closely related results in the in vivo model as well, like the in vitro assays. Thank you. Okay, this is easier.
She has engaged in the research of R&D of herbal medicine and development of animal medicine. She targeted for neuropsychological diseases. And she has over 30 articles in international journals. I invited her to start the presentation.
Sorry, there have some mistakes. Please wait a minute. So sorry. Now I am start. Ladies and gentlemen, good afternoon. I'm honored and proud to have the opportunity to speak at this meeting. Today, I would like to present my paper. Jensen said RB1 produced antidepressant-like effects in a chronic social defeat stress model of depression through the BDNF-TRKB signaling pathway. I am Jiang from the Peking Union Medical College, Beijing in China. Now I will introduce my paper from the 4 parts: background, materials and methods, results and summary. As we all know, depression is a common severe and life-threatening mental illness. And in 2014, about 350 million people of all ages suffered from depression. And a report from the WHO reported that by 2020, depression is become the second most burdensome disease for human beings. Now, main drug used for depression treatment is more based on the menomine hypothesis and worked increasing the levels of neurotransmitters, included SSRIs, NARIS, and SNRIS, and most of them only have effective in about 1/3 of patients and show serious side effects. So there is a need to explore the more effective agents for depression and cause fewer side effects. The mechanism of depression is not fully understood. In the past decades, several hypotheses such as the neurotransmitter hypothesis, neuroplasticity hypothesis, and so on. Recently, the BDNF signaling pathway is demonstrated played a crucial role in depression treatment. BDNF, which is one of the most important targets in depression treatment, has been demonstrated to participate in hippocampal neurogenesis. Previous have showed that the levels of BDNF was decreased in the hippocampus of stressed mice, and while chronic administration with antidepressant drug increased the BDNF expression levels, followed by the decrease in neurogenesis. As such, the regulation of BDNF signaling pathway and hippocampal neurogenesis may provide a new approach to treatment of depression. The herbal medicine have been used for many years and have been proved effective for treatment of depression. The ginseng is the most popular traditional herb used in Asia, and RB1 is the most active components from the ginseng. And many studies have studied RB1 and found they have numerous effects such as anti-tumor and antioxidant and anti-inflammatory effects. Recently, publication reported that RB1 have a prominent antidepressant effect. They could improve the depression-like behavior in chronic unpredicted mild stress model and CRS mice model. And in this study, we was aimed to investigate the antidepressant-like activity of RBE on the CSDS mice, and further explore the possible mechanism and the role of BDNF signaling cascade in the hippocampus. The materials and the methods. In this study, we use animals include the CD1 mice and C15-7BL mice, and the drug is RB1 and IMI as the positive drug. The model is CSDS model. We study include 2 experiments. Firstly, we split into the mice for 5 groups, and to study the antidepressant effect of RB1. Then we used the K252A as an which is a effective TRKB inhibitor to examine the function of BDNF TRKB pathway in the antidepressant activity of RB1. This is the experimental design, as we can see in the picture, except for the control group. The other mice was exposed to the CSDS procedure for 28 days, and the behavior test include the social interaction test, sucrose preference test, and forced swim test. The CSDS model, which is associated with the depression in human and simulated human emotions, such as fear caused by failure and frustration. In this model, animals were subject to psychological social stress through a stress procedure with excellent face and constructive and predictive. In this model, the C57BL mice were subject to physical defeat by exposing them in a CD1 mice for 5 minutes daily. And then we used a plant to celebrate the 2 mice. The following day, the test mice is the 2 mice was exposed to the chronic psychological stress for the subsequent 24 hours. The behavior test. First, we conduct the social interaction test. The test was include two phases. The adoption period without interactive target in the arrow, and then a CD1 mice was put in the arrow. The C57 mice maybe show the social avoidance behavior as the time spent in the interaction was decreased. And the sucrose preference test, when the mice were it is a classic behavior test for test the depression behavior. When the mice was anhedonia, the sucrose consumption was significantly decreased. In the first swim test, the mice will show the behavior despair, as shown by the immobility time was markedly longer. Then the biochemical analysis include the WB and IF test. We mainly test the BDNF signaling cascade, including test the ERK and CREB, AKT, and BDNF. And the IF will test the hippocampal neurogenesis as tested by the NEUN and DCX double labeling experiment. The statistic analysis was one-way ANOVA and two-way ANOVA. the results in the experiment 1, the RBE improved the CSDS-induced depressive behavior in the mice. We can see from the picture, after 4 weeks of CSDS-induced a depressive-like state, including the anhedonia, social avoidance, and despair behavior, as evidenced by the decreased amount of sucrose solution consumed in SPT, and the increased time in the interaction though in SIT, and the increase in the immobility time in the FST. These behavior changes were significantly improved by long-term RBE administration. And DCX is a mirror tube-associated protein that was expressed in the newborn neurons, and it as a marker of neurogenesis. We can see in the picture, the CSDS mice showed a marked reduction in number of the DCX cells in the DJ. Treatment with RBE significantly increased the numbers. And in the WB test, we found this similar results. A significantly decline in DCX protein expression in the hippocampus of the CSDS mice, whereas treatment with RB1 was increased the BDNF expression. So the results showed that RB treatment reversed the CSDS-induced decline in the hippocampal neurogenesis. The WB result in the BDNF signaling cascade showed that BDNF protein expression was increased by the RB treatment, and RB treatment could evaluate the levels of the protein expression of ERK, CREB, and AKT, which have been associated with activation of BDNF signaling way. The results showed that RBE treatment reversed the CSDS-induced inhibition of BDNF signaling cascade. Then we co-injected with the CSDS mice with the highest dose of RBE and the K252A, which is an effective TRKB inhibitor to access the essential role of BDNF TRKB signaling pathway in the RBE. We can see from the behavioral tests, the K252A pre-treatment abolished the RBE-induced decrease in the performance in the behaviors. And the IF results also show that K252A blocked the effect of RBE on neurogenesis based on the reduced number of DCX cells in the DJ of the CSDS exposed mice, which is rarely showed that BDNF is necessary for the RBE-induced antidepressant effects. The Western blot results showed that K252A prevents the effects of RB1 on the expression of BDNF, PCREB, PERK, and PAKT in the hippocampus of the CSDS exposed mice. Our results showed that RB1 prevents the depression-like behaviors in the CSDS mice, and the antidepressant effects of RB1 require the BDNF signaling way. These results showed that we can say RB is a potentially effective neuroprotective agent for the treating depression. This is the finding. Thank you for your listening.
We asked Dr. Ryan, Assistant Professor, to start the presentation. And the molecule mechanism. Dr. Ryan is graduated at 2019 and obtained a PhD degree in pharmacology. Her main research focuses on screening national compounds, finds potential influence of the treatment of osteoarthritis diseases. Some research findings of her are published in modern
This mic is here and they cannot hear it. Just stand here and solve the problem.
We asked Dr. Roaniton to come and start the presentation. Dr. Roaniton is Assistant Professor. The title of presentation is Effects of Sesquiterpenes on Osteoclast and the Molecular Mechanism. Dr. Roaniton graduated at 2019 and obtained her PhD degree in pharmacology. Her main research interest focuses on screening natural compounds to find potential chemicals for the treatment of osteolytic diseases. She has published his article, an article in the famous journals like British Journal of Pharmacology, GMP, Journal of Natural Products, and Frontiers in Pharmacology. We ask her to start the presentation.
Dr. Tan, can you hear us? Dr. Tan, can you hear us? Chat खोलो ये ना chat problem. Next one. ये हैं ये जी बोलते कैसे बोलते हैं और आप जैन देखिए नहीं वो भी नहीं Dr. Yi Zing Q, can you hear us?
Yes.
Okay, so I think we'll have to take your presentation if you're ready. And if you are, we'll ask the chair to introduce you.
Okay.
Okay, just give us a minute. The chair will introduce you, and then you can start your presentation. So if you could please come
Okay,
here.
thank you.
Use the third presentation. Yeah. Okay. Okay, you can start your presentation. If you would share your screen and start the presentation, we are ready for it.
Okay, let me share my screen. Can you see my screen?
Yes.
Okay, thank you. Good afternoon, everyone. First, I wanna express my appreciation for organizers' invitation. Thanks for giving me this chance to share my research. My topic is DNA enzyme and RGO-based fluorescence assay for FPG activity analysis, drug screening, and bacterial imaging. My presentation includes 4 parts: introduction, experimental design, results, and conclusion. The first is introduction. When DNA exposure to ROS, the guanine was easy to be oxidized to produce 8-oxyguanine. If unpaired, can lead to mutagenesis through GC to TA transversion during DNA replication. This mutagenesis has close relationship with the onset of some tumors and inflammation. The enzyme FPG
इनका नेट का है ये देखिए इनका नेट
Next is the sensitivity and specificity analysis. For the sensitivity analysis, the detection limits was lower or comparative to the recently reported methods. To confirm the selectivity of the method's other 5 enzymes and the protein BSA were examined. But there was no obvious fluorescent enhancement after the addition of other enzymes and protein. This figure indicates the sensitivity, the selectivity of this biosensor is good. Then the developed biosensor was applied for the compounds screening. 11 natural compounds extracted from TCM or ethnomedicine were screened, and 7 compounds were identified as activator. Among these compounds, the compound G, glycinoside RE showed the strongest
activation to FPG. This part is the activation effects of genocidal RE. Next, we did the computer docking to explain the interaction between FPG and the compound G, genocidal RE, and we find the benzoyl ring of compound G interacted with the Hayes E127 of FPG through pi quotient. And the E score of compound G to FPG was very low. To further expand the application of this biosensor, we tried the FPG imaging in E-Coli. First, we optimized the concentration of the added biosensor and the co-culture time of the biosensor and E-Coli. We found that the compensation of this system consists of 10 microgram milliliter RGO, 150 nanomole, and 120 microgram per milliliter of RGO. Could meet the requirement of the detection. In addition, the maximal signal intensity reached at 4 hours. This one. We also studied whether the compound G, genocidal RE, could activate FPG in living cell. Through the imaging, we found the genocidal RE also could activate FPG in E-Coli. This is the statistic results. Because FPG has close relationship with ROS, we further test the effect of compound G on ROS. The ROS imaging results indicated genocidal RE also could reduce ROS in E-Coli. Conclusion. That's the whole story of this work. Conclusion is the last part. In this work, a sensitive, simple, and reliable fluorescent biosensor was developed by taking the advantages of DNA-assisted cascade signal amplification and ultra-high fluorescence quenching effect of RGO. With the help of this biosensor, 11 natural compounds from 2-Jia Ethylene Medicine and TCM were screened, and 7 compounds were identified as activator of FPG. More importantly, the developed biosensor can be used to visualize the activity of FPG in E-Coli, which is the first study of fluorescent method for FPG imaging in living bacteria. The imaging result visually demonstrated the FPG activation effect of genocidal RE in E-Coli. Meanwhile, the scavenging effect of genocidal RE on ROS in E-Coli was reviewed, indicating that genocidal RE has oxidative damage repairing effect. This work was conducted under the guide of Director Professor Wang Wei in Hunan University of Chinese Medicine and Professor Liu Bing at Hunan University. Thanks for their help and other group members' help. And finally, thanks for your attention. That's all for my presentation. Thank you.
Thank you very much. Very nice presentation, a new method by analytical chemistry and making a new proof by quenching the fluorescence to quantify very large quantity of fluorescence, even less than nanomolar, especially in living bacteria. Thank you very much. Is there any question? So, thank you very much for your nice presentation. Now you can proceed for tea break, and also after the poster session is held at H.E.J. Lounge. Yes, we can go all together. Yes, thank you very much.
Thank you.